So I'm going to do a little backtracking here. Every time I find something, I try to update if it looks interesting. So we've been talking a lot, or at least in the past, we were talking a lot about helicases, these 6-member ring helicases like DNA-B, MCM 2 through 7. This morning, it's just published. It's still smoking hot. Nature Structural Biology, the new edition for this month, came out, and there was an article in there, a review article on the ring helicases and a mechanism, structure and mechanism on how they work. So since I'm interested, you are interested. Let's look at how this works here. What you'll see is that it's a 6-membered ring, and each of these binds ATP and burns ATP. We sort of knew that. Every advance in a base pair that it breaks, as it breaks the helix, it has to burn an ATP. And here you see the bacterial configuration and the eukaryotic configuration. This would be the MCM 2 through 7. There are two, it looks like this is a double helicase, but actually they're just 2 domains, an N-terminal and C-terminal domain. So that's it, that sort of knob on the back is part of the same subunit. But I thought it was interesting that in the C-terminal domain, which is this one that's leading its way, probing into the double helix to melt it apart, look what you have. You have a Rec-A fold. A fold is what structural biologists call a domain of a protein. A histone fold. It's not actually a fold, it's not actually a cleft. It's a configuration of helices and beta sheets and so forth that show homology or serve a function. So it's called a fold. Anyway, there's a region in there that is very similar to Rec-A. Now, Rec-A has learned to manipulate single-stranded DNA and make it look like and fool it into thinking it's still configured in a double-strand helix, even though it is displayed on this scaffold as a single strand. And you might think how that could be useful as you're trying to provide a site. You're trying to coax the 2 strands apart. You got to go, look, it's almost the same. You can just find over here with me, baby, and forget that other fella, that your complementary strand, because I have a scaffold here that you can land on and you'll hardly notice the difference. It'll be just like you're double-stranded. Doesn't last long. It quickly moves on, and now it can never find its previous partner, and it just has to, don't look back. But you can see the emergence here. Probably the helicase learned how to do it first, and then that became Rec-A, but who knows the evolution or sequence here. And the other thing is this ratchet mechanism of how it actually moves forward. You have this ring, and notice they are it's sort of like, for those who work on bicycles and stuff, it's a split ring. It's a ring with a cut, and the 2 ends are not even anymore. One is up above the other one, so that when you tighten down, you'll get some back resistance. It's a split ring here, you see, and what happens is it burns ATP, and the subunits slide past each other into a forward position. See, this is the original position. The one that's right under the fork, which is B, moves up, and as it moves up, it melts a base pair. And then the next one in sequence moves up, which will be C, and that melts, and then D, and then E, and it just keeps going around that way. The one beside it moves up, then it's turned to move up, and just ratchets up. And the difference between us and bacteria is that our helicase is on the template for leading strand. Okay, so I thought that was an interesting aside. Now that so you can be properly educated. let's jump back into transposable elements, and we're still on part 1. And these are ones that I've taken the blue out, which is, it seems like it should be trivial, but it takes for hours and hours to do it. Okay, let's see, I think we were right pretty close to here. This is kind of an overlap slide. Security alert, help protect me, I don't know. Okay, I'm okay really, you can stay back there. So, you know, when they intervene in those prison channels, MSNBC, they play prison stuff all day until Rachel Maddow comes on. They go in the cell and they got somebody and they come in. So anyway, I digress. Transposable elements. We were talking about composite ones. We talked about the simplest transposon is an IS, insertion element. It is simply an inverted repeat at each end, and there's an open reading frame that encodes a transposase. That is a functional one. It doesn't need, it's all self-contained, it's autonomous, it can move itself. If you have the inside region, the transposase, mutated, it can still move, but it needs a helper transposon. You see this reoccurring theme? All you need is a cut site, an ORAT, and something to help. The helper plasmid, agrobacterium, has used that strategy. Here you see composite transposons use the same strategy. The idea of you just need the cut site, and then you can mobilize whole armies of DNA to get on the move, and we'll see how that might work in a minute. So composite transposons have stuff in the middle of two copies of the same transposon. They are called arms. So this is IS50. There are 2 copies. The orientation of the arms doesn't matter. They could be inverted repeats of each other or direct repeats of each other. It really doesn't matter. In order to move as a unit, you're going to have to cut on the outside extreme inverted repeats. You can't cut on the internal ones because then you would cut the individual ones out. Okay, so quite often what you'll find in between the 2 arms are things that can be easily selected for, either in the wild, in the sort of natural context, or in maybe a medical context, or a lab. Antibiotic resistance. Here you see a variety of them. This is a well-studied one here, IS-10. IS-10 on either side. This makes transposon 10. So we'll see what the numbering looks like in a minute. Let's see what the verbiage is here. So it's not changing slides. It's always cool. Okay. Central region can carry drug resistance. We can carry anything from the chromosome. And the flanking IS elements can be in either orientation. Okay, this is a figure from the book. And here you see again, just drawn a different way. I see. The battery's going dead on this. That's the problem here. You got 2 of them? Got all this electronic stuff going on.
No, but the part that plug the other part in?
Replug the thing here, maybe? Oh, yes, it did. It's an old man thing. Well, look at that. Okay. And maybe teach one more semester before they push me out the door. Okay, where were we here at this exciting lecture? IS elements only carry the enzyme needed for transposition. So it codes transposase, or it could have a transposase and a resolvase if it was a replicative IS element, right? But the IS element itself is not what carries the antibiotic resistance. The antibiotic resistance is in a composite transposon. It's always intervening material between the two IS elements that comprise the composite transposon. Notice the way they are named. If it's an IS element, it's IS and then a number. If it's a composite transposon, it's given the designator TN for transposon. TN10 carries TET resistance in between the two arms. Usually only one arm is functional, which means only one arm has an active transposase, the other one is full of mutations. All you need is one. One sort of acts as a helper for the other. The only two, what's an IR? Inverted repeats. The only two IRs are needed. There are four here designated by the red arrow. You need the outside ones only in order to transfer as a unit. In fact, you don't want the inside ones to be active. Okay, and sometimes that principle appears on homework questions and on exams. Which of the four inverted repeats can be dysfunctional or are dysfunctional in order to have the unit always act as a composite transposon? In order for it to always act as a composite transposon, you're going to have the two outside ones active and the two inside ones mutated. Of course, in some circumstances, it will not be 100% deactivated. You can have circumstances where it's 50% mutated in terms of function, and we'll see what consequences that may have. In fact, you'll see it here. So here you have, this must be TN10 because you have TET resistance in the middle here. Here's the transposon. Here's the transposase coding region. And here are the two inverted repeats. And IS10 left and IS10 right. And this is the plasmid target that they're going to go into. It integrates into that. And now that it's in, the next time it moves, there are two possibilities. Two possible outcomes. And just think about this. If it cuts like it did originally on the outside here, it'll have the same kind of jump. But let's say the inside inverted repeat still is not very mutated, just slightly mutated, and sometimes it is able to be cut. If it happens to cut on the inside ones, then the thing that jumps will be everything except for the transposon, right? So the two possible outcomes reflect that situation. You can have the original transposon, TN10, jumping if it's cut on the outsides, but if it cuts on the inside, notice how that is really the inside of the composite transposon, then the thing that moves will be the original target plasmid itself will get incorporated into a second molecule. So you can see the outcome depends on which pair of inverted repeats it uses on each of the arms. And this reflects sort of a evolutionarily situation that's in between the two possibilities, because sometimes there's enough homology for it to cut on the outside ones, and other times, just by probability, it will cut to these ones that are mutated on the inside. Everybody understands that concept? Okay. Summary so far. Smallest transposons are called insertion sequences, IS elements. These have inverted repeats at either end and a transposase gene in between the inverted repeats. They do not carry resistance or other kind of markers, that's only a composite transposon. The two IS elements flanking marker genes form a composite transposon. The IS elements at each end are called arms. The frequency of transposition for composite transposons is not as efficient as for an IS element itself, because the second site it has to cut is further removed. So if you get just a few transposases produced in the cell, the probability of it cutting a second time decreases as that composite transposon gets larger and larger. Just because it's further away from the first cut, you reduce the probability that it's going to find the second site. So the next topic. This is related to transposition, except this is deceptive. It's not really. This is back to homologous recombination, but between identical IS elements. You remember we said that the F plasmid, or many plasmids, will integrate into the host bacterial genome if they have the same insertion sequences that the host genome has. That provides a basis for common homology, which of course now is a substrate acted on by the homologous recombination system. So this is the outcome. There are two outcomes here. One, if the IS elements themselves, of course these have to be the same IS element. This is the kind of configuration that you would have on a composite transposon, for example. But it could just be the same, two copies of the same IS element. Each of these is perfectly functional, and it always just cuts itself out. It doesn't move as a composite at all. But it could be that situation, or this could represent a composite transposon. The functionality of the transposon itself is irrelevant, because this is what the host does just because it's homologous, two copies of the same DNA. It's homologous recombination. So if the two IS elements are in direct repeat of each other, notice they each contain within them inverted repeats, these little red arrows, but the entire IS element is in direct repeat with a second copy of itself. Then in order for it to line up with homologous recombination, you're going to have to do this little flip of the strand. And you can connect this in your mind with homologous recombination. It would go purple, crossing over back into purple. And the other way to look at it is light blue crossing over into light blue. So you'd get this happen. You would get the intervening sequences cut out. How do you remember that? It's easier to remember the inverted situation, but we'll get to that next. I remember this as saying direct repeats lead to direct excision. Excision if they are direct repeats. Reciprocal recombination, direct repeats result in excision of anything between the two. This may be on homework. It's often actually on the exam. Who cares about homework? This thing could be on the exam. Just saying. This is the other possibility, that you have in the genome someplace or on a plasmid. Homologous recombination will happen wherever there are repeated sequences in the bacteria, whether they're on a plasmid or in the bacterial genome. If the IS elements that are close to one another are inverted, then when they line up, as you can see, dark blue going in, crossing over here into the herringbone pattern, then crossing back, it would end up flipping or inverting the intervening sequences between the two identical IS elements. So this is the easy one to remember. If you have inverted IS elements, the material between the two is inverted as a result of homologous recombination. So that's the one you should stick in your head, and just know the other one's the opposite, that it's cut out. Okay, so the next series of slides that will sort of take us out of this transposons part one, where we're looking at the detailed mechanism, we'll talk about the two general types of jumping, of transposition. The transposition that does not make a copy of itself in the jump, non-reciprocal transposition, and then we'll compare that to reciprocal transposition, where you leave a copy from the place of origin. So here you see non-reciprocal transposition. It leaves a big hole, and what else does it leave? It leaves a direct repeat, right? This was the original target. It had no homology at all with the IS element or the transposon, none whatsoever. It was chosen at random. This little thing, of course, was not repeated, just happens to be any 9 bases, but because when it went in, due to the repair of the staggered end, it's going to make that sequence repeated. And when it jumps out and gets put back together through non-homologous end joining, that extra copy is going to still be there. Now, if it doesn't disrupt the reading frame, you have a possibility that it will be benign and have no phenotypic consequence. But quite often, it's not in the same reading frame, and if it is, it could be in an active site. It would end up still being mutated. You would never know that it jumped out. Because if it does revert, it's a low event you see here. It depends on the transposon, but for TN5, it's what, 1 in 100,000. If it's TN10, it's 1 in a billion chance that it will heal itself when it jumps out, because it has to get rid of one of the extra copies of that little direct repeat caused by the original insertion event. That's not very likely, but it does happen. It probably involves a recombination mechanism. Okay, so that's kind of an overview there of non-replicative transposition. Let's go and look at some of the issues or complications of replicative transposition. It proceeds through a co-integrate structure. Only replicative transposition has this, where before it gets resolved, you have both the target and the donor attached to each other in this large co-integrate molecule. Replicative transposition encodes not only a transposase, but a resolvase to, by site-specific homologous recombination, resolve these two DNAs into their original separate forms. Creates a co-integrate structure. How is it resolved? And it's resolved, let's see here, what's that change? Recombination between the two copies, at a particular site. It's not just homologous recombination. This resolution is mediated by a transposon encoded open reading frame, a protein, resolvase. And it acts at a specific site. So this is not general recombination. It is homologous, but site-specific. This little site, RES, which nearly always appears on an exam question. RES is the site, and it's located within the transposon itself. That's the region that actually crosses over using the resolvase to separate the two DNAs. Okay, so for years this used to give me fits. Now we're going to get into the actual how the strands go. And I'm a little bit dyslexic, and trying to look at where the strands are and keep track of it. It was only this morning, this is a happy day, that I finally got it straight in my tiny head. And so I'm going to share the joy. I'm going to show you guys so you don't have to spend your life worrying about this, and you're forced to make little things in clay deep into the early hours of the morning. You'll thank me many times. Okay, So you've got a pink and a blue molecule. I got that so far. Boy, girl, I guess, I don't know. The pink one is the donor. Even that takes a while to wrap your head around. The pink one is the donor, and notice there are cuts all over the place, but there seem to be, we'll talk about that later. We'll get the major points here. Strand transfer complex. This is the thing that, if you don't watch it, leads to a co-integrant. Notice what's happening here. You are getting the donor, the pink, actually being physically attached to the target. And then you're getting 3' ends generated, which act as primers, so you're getting sort of a repair DNA replication event that comes afterwards. So you've got some 3' ends here that are left over, and these 3' ends are on the target molecule. Let's get a little more. We're going to have another slide that gives you more detail on this, I think. Is that going to be the next slide? I'm not sure. Maybe this is not my latest version, but I should. Oh, yeah, I wrote it up there. Okay, so this is what happens. Notice the figure here is a little misleading. This line here is black. This is the donor. It should continue to be black. And notice it just mysteriously joins the target. There are two kinds of 3' end activities here. The donor, the end of the transposon itself, attacks the target randomly and breaks the sequence and ligates itself randomly to a nucleotide in the target. The 3' end does that on each side of the transposon. So let's see if we can see this. That 3' end attacks and covalently joins itself to the target. And the other, that's one, and then the other side of the donor is this line, which should be in black. That physically attacks the other side of the cut and joins the target. So the two 3' ends that are coming from the donor cannot be used as primers, can they? Because they are already used up in this sort of strand invasion where it invades and ligates itself, joins itself to a nucleotide in the target as if it's the same DNA strand. It can't be used as target. So the 3' ends that are left, here's one, and there's the other. They now are, no, excuse me. Here's one, here, and here's the other. These 3' ends in the target, here and here, act as primers for DNA synthesis. And when that happens, you're starting in the target and you're going and using as template the transposon itself from the donor. And then you're going, continuing around the circle, and this is how it becomes a co-integrant. On the other strand, it starts here. This is a 3' end in the target. It's elongated, and as it's elongating here, it's making a copy of the other strand of the transposon. And it continues over and now copies the other strand of the donor. And that's the origin of the co-integrant. It's a result of DNA synthesis that originates from the target as a primer. But all 3 heads have exploded. I need to change the slide quick just for safety reasons. Replicative transposition. Formation of the co-integrant. Here you see it, and here you see that little pesky exam question. What is the site of that resolving? It's at a RES site. That's where the resolvase, encoded by replicative transposons, that's where it acts and performs a site-specific homologous recombination. So you see how all these subjects are interrelated. We started with DNA replication, then recombination, and transposition. They all sort of use parts of each other's pathway. And so what you end up with is an additional copy of the transposon after it's been resolved here. Replicative transposition. And this is from molecular biology of the gene. This is where I learned that it's the 3' end that does, from the transposon itself, that does the attacking. This is where that model is shown, and so I've incorporated that from molecular biology of the gene to illustrate this replicative transposition involves attack of the 3' end of the target molecule to the target, of the should-be donor molecule to the target. I need to edit that. Cuts and joins donor at both ends of the transposon. This is unclear language. It cuts and joins the donor to the target at both ends of the transposon. So that's going to take a little editing. Second, the replication fork forms at one of the ends and extends the 3' end of the target for leading and lagging strand synthesis. So here you see it goes in, and you see the connection there. The transposon is directly connected to the target, and that's a 3' end on the on the target left. And then on this side, it's the other strand of the transposon that joins itself using its 3' end to the target. And you have two gaps here. You have the potential to form two replication forks. What happens is only one replication fork is formed at either of the two ends, and it's leading and lagging strand, just like replicating the genome. It happens right there. One extends the 3' end of the target, which is leading strand, and then as that replication fork moves, it lays down the second strand, lays down the lagging strand, using which is now a very familiar mechanism to you. Here you see non-replicative transposition. You know, the most difficult is the replicative. Non-replicative has two mechanisms. And it depends on when you do the cuts. The outcome is the same, that it gets totally cut out of the donor and inserted into the target, but there are two molecular pathways that it could follow to achieve that. So transposon is excised totally free. See, it's cut on both strands, totally free, and then you get the 3' end of the transposon attacking and physically joining to the target each of those 3' ends of the transposon, leaving once again a 3' end on either side for, in this case, simply repair. This is just a little gap, and on each side it's just filled in. DNA Pol 1 can do that. It doesn't go all the way around and make another copy of itself. There's nothing being copied except just a little repair operation here and there. And of course, it leaves a big hole where it came from. Here you see the more details on the mechanism of this attack by the 3' end of the transposon, how it can actually cut, and at the cut site it makes, because the transposase itself is bound here. It mediates this reaction, the transposase, cuts and ligates the 3' end to the cut at the target site. Its 3' end joins, of course, the 5' end of the cut. Joining the 5' end of a cut, doesn't that sound familiar? Okay, so there are two ways you can can do this non-replicative. It seems like it should be so simple, but this is as complicated as this gets after these slides here, learning the mechanism of replicative and non-replicative. So here you have the non-replicated mechanism called breakage and reunion. I haven't figured out a way to have a little way to memorize which is which yet, but I'm working on it. I still got years and years to figure this out. A strand transfer complex in this mechanism is a part of the pathway. Remember, strand transfer complex was certainly an intermediate step in replicative transposition. So non-replicative and replicative share an intermediate, at least one of the pathways of non-replicative transposition does. And this is the one, breakage and reunion. So you see the attack here, it joins, the end of the transposon actually attacks and joins at the cut site of the target there, and you see it's joined there also. And then you get a second cut, which we'll see in a minute. The cuts occur. So the first cut is what the transposon does as it attacks, cuts, and ligates. And then the second cut occurs on the remaining strand of the donor, and that second cut now is what frees it up so that you end up with a big hole in the donor and transposition of the transposon in one copy. Non-replicative transposition occurs if the crossover complex is nicked at the unbroken pair of donor strands. The first is where the transposase attacks the target and joins the 3' end, so that's a cut, to the target, and then you have a remaining strand on your donor, which is pink, that has to be cut in sequence. Breakage and reunion. How is that different than cleavage and ligation? Well, cleavage and ligation is simpler. What you get is, before there's a transfer, you get both strands cut on either side of the transposon. It's totally cut out before it begins to search out the target and insert itself. That's the only difference. So let's look at this mechanism also used by many eukaryotic transposons. Cleavage and ligation, let's go back and see how this differs. You notice here you get this joining attack before you cut the second strand. Breakage and reunion. In cleavage and ligation, both strands are completely cut, and then you do the attack. That's the difference. So this one does not have a strand transfer complex. Cut and paste is another name for cleavage or cut and ligation, paste. And then you see some of the details of mechanism that we've learned by looking at some of the specific transposases. And you can see the nature of how can you cut out, this is just the molecular steps. You will not need to know this level of detail on the exam, but just shows how a double strand cut was made. This is more detail on the transposase itself, and this has relevance because this explains why the cut is staggered. It's because the transposase has two subunits, each able to cut. And they cut They can't occupy the same space, so they cut at different sites because there are two subunits. And the topology, or the physical dimensions of this transposase itself, determine how close those cuts can be. That creates the stagger, and of course the stagger in the cut, or how much they're offset, determines the length of the direct repeat that is generated in repair of that overhang event. Okay, so you see here that the transposase has two subunits. Each is involved in a cut. So the physical geometry of the enzyme itself determines how close you can smash those two subunits together. And that, in turn, determines how much offset there is in the two cut sites. If you have two great, big, fat transposases, they can't get too close together, so they have a really big offset in where they cut.
Okay.
It would generate a larger direct repeat at the target. Okay? That's the end of Transposons Part 1. That explains the mechanism down at probably more than you'd like to go, but that's a pretty comprehensive coverage of the basics of mechanism, looking at bacteria as I guess a model to inform us. How are we doing for time? Time for a break? Okay, let's take a 5-minute break, and then we'll go into how transposons work in eukaryotes. So this is sort of genetic engineering, right? This took thousands of years. We can do the same thing with a lot greater precision, and it may take us 5 years. But of course, for some reason, apparently the public thinks it's unethical to do it in 5 years when it should be taking 5,000 years. So anyway, I let my emotions show there. Okay, transposable elements in eukaryotes. These still have the original blue background, so for the next couple of days I'll be taking the blue out, removing all the shadows, so it'll be more easy to print these. Eukaryotes have the same classes of transposons bacteria do in terms of replicative, non-replicative, DNA-based transposons. They encode resolvase and transposase and all that stuff. It's the same principle. They arose at the same time. We inherited them. We also have a class that's not much found in bacteria, which are the retroviruses, which we'll talk about a little bit later here. But here you see examples of the effects of transposition. You see here the corn, and over here, what you'll see is the original color of maize or corn was this dark purple color. And when transposons jumped into the gene that makes one enzyme in the pathway for anthocyanin, it removed that pigment, and what you see are the other pigments shining through, predominantly yellow. So what we normally eat is a result of that, a mutant. It probably has multiple transposons in it now, and it's not likely to jump back out again. But corn is full of transposons. More than half the genome is just jumping around. That could be one of the reasons that it was so, I guess, malleable to breeding, to changing from teosinte, which was just ridiculous, to what we see now as maize. Also, you can see this eye color thing going on in Drosophila, the fruit fly. The original color, like corn, of the eye is red. And what you'll see is you'll get a transposon jumping, and then you'll begin to get it jumping back out again, reverting. When it jumps back out again, if it does it really cleanly, it's a bright red, but if it's at a location that when it comes back out is not a precise reversion, it still has mutant properties, and so it's not quite as intense as it originally was. In some places it is as intense. When you see this happening in maize, what you see here, these little sectors or chimeras, these are all cells that are derived from the same cell as it divided. And in these cases, the transposon jumped out, and now you get a restoration of the color. And so you can tell when this happened in corn development, and development of this particular ear, by how big that spot is. If it happened more recently in the development of this one kernel, then you get little tiny speckles. If it happened very early, it could cover half the kernel. If it happened really early in the germline, you'll see complete reversion of the kernel, like you see up there. If you look at some of this stuff that I grew in the backyard, you can see some jumping around. These, of course, completely cured. And then there are others with sectors. You see these streaks? Streaks where it jumped out, reverted. Here are big sectors. Very early in development of individual kernels. This is called Cherokee flint. I got it from a farmer in, I guess, Archer, and he got it from a farmer who was of Cherokee descent. So this is like right straight from the guys who invented it. Native American biotech. Got it right back from the source here. This only mules like. It'll break your teeth. It's really hard. That's why you chew it up and make it into a corn paste. You let the stone do the chewing instead of your molars. The more modern breeds are soft and plump and easier to eat. But you'll also see in here reversions that affect things other than pigmentation. It's hard to see here, but this white is a buildup of starch. You have a missing gene that's this conversion from starch to sucrose. It's mutated, so you get accumulation of starch. It doesn't get turned into the juicy sweetness of sweet corn. That, all the genes of corn, of course, are subject to this invasion by transposons. We can only see the ones that are easy for us to detect. Pigment. Okay, if I can figure out, how do we do this, Lance?
Power button on the red thing.
That's all.
And then that,
Okay, I gotta eat my Wheaties.
Yeah, you get that.
Didn't take but 2 thumbs.
And then that.
Okay.
And then
Thanks, that young man.
which one of those?
One of those. Okay. Oh, we got.
No, hold on.
We got.
That
We're
one.
like hitting on one cylinder. You hadn't heard that in a while.
Oh, I can't see these. What is that?
It's computer. Is that?
No.
Whoa.
There you go.
There you go. Still got it. Okay. All this will get edited out, of course, because I do want to keep my job. This is what I just talked about. You see where you get these chimeras, you get a jumping out, a reversion, then you get either a white spot or a region with color. Controlling elements in certain extra genes with visible phenotypes. And heterozygote, mitotic descendants give rise to a sector. This is called a sector. And this change in phenotype is called variegation. I don't think these words are on exams. Variegation can result from insertions, deletion, chromosome breakage, and rearrangement. So that's one of the big problems with transposons, is that these little color changes and little minor things, they're not very disruptive. But if you lose an entire chromosomal arm, it's bad news. In maize, DS elements are in particular prone to causing chromosomal breakage. So let's look at the system here in a minute. Ear of corn with multicolored kernels caused by transposable element DS. When DS inserts in a gene required for kernel color, it disrupts the gene's function, resulting in colorless, they're really pigments there. When another transposable element AC is present, the DS element jumps away, restoring gene function, which is back to red-purple. The multicolored pattern reflects both when and how often DS jumps. And we're going to learn about these two transposons in maize in the next slide here, I think. Nope, we're going to talk about the person who first discovered it. You know, we say transposons. The word first was formed in the buccal cavity of this lady. Transposons, she said. She looked at it and it came out. Boy, that was fun. Transposons. She named it. She discovered this before we knew the structure of DNA. Barbara McClintock. She was actually at Cold Spring Harbor when I went there one time. I took my graduate student who was quite, you know, she could talk to anybody. And Barbara McClintock was sitting, they had a little cabin for Barbara McClintock. She was like a national treasure. She could stay there at Cold Spring Harbor as long as she wanted to. She figured this out in the 40s, and they didn't, you know, Watson Crick and, what's her name, Rosalind. What's her last name?
Franklin.
All the women said Franklin. Indeed, yes, Rosalind Franklin. When they finally worked out the structure of DNA, she knew that genes were jumping and were mobile and were transposing from just looking at these kernels of corn. She was kind of a recluse. She didn't like to talk to grown-ups, but she talked to graduate students. And so she talked to my graduate students. She was sitting on the steps of a little cabin watching people at a conference, you know. And my student went over and sat down with them. They had a nice little conversation. She would smoke a corncob pipe, wore khaki pants, and was quite a character, but very reclusive. Barbara McClintock won the Nobel Prize in '83 for physiology and medicine. Half of our genome is composed of transposons. Anyway, I thought I'd give a plug for women in science there, but I don't get any points because I couldn't remember Franklin. Okay. Maize, DS. We have these two, well, it's really, it used to be one. This transposon, it's a DNA-based transposon that one copy has lost its ability to jump on its own because its transposase is so sorry, it's got so many mutations that there's not much left of it. But it's still got the cut sites. The original transposon, called AC, still has a functional transposase, at least lots of copies. Even some of that has dysfunctional transposase. But anybody who's got a transposase that works serves as a helper to anybody else who has an ORAT. I mean, anybody else who has an inverted repeat, right? It's the same principle. Autonomous and non-autonomous transposon. The autonomous ones are like helper plasmids, except they do contain their ORATs. They do have the cut sites, their inverted repeat. They can move themselves. But they can move anybody in the family who still has retained the inverted repeats. And sometimes because they can move and replicate, who needs a transposase? And so they'll get smaller and smaller, and some of them will be as small as 300 to 500 base pairs. It's just 2 cut sites. It's like left and right border. It's whittled itself down into a small mobile element called DS, and there are many varieties of it. But some of these are so small, they're called mites. They don't code for anything, they're very small, and they're highly repetitive. Hundreds of thousands can accumulate in genomes. We have repetitive sequences just like that. Primates do. The ALU1 repeats. ALU is a restriction enzyme. They found that if they cut the genome with that, you'd get these bands. And so the repeat regions, the transposon infested areas, have their own density if you did a cesium chloride type of gradient. And you'd see it there as a satellite peak. So they were called satellites. Anyway, transposons. So this is the harm they can do. Maze DS stands for dissociation. It causes especially high frequencies of breakage. You know, when it jumps, it leaves a hole. That's why it breaks. It doesn't get repaired fast enough. And you can lose your arm. But, well, you're losing your arm because why did you lose it? Because only one of the resulting fragments is going to have a centromere to be segregated between the 2 daughter cells. That's why you lose it. Acentric fragments. That's an acentric fragment. That's DS. And so what you'll end up with, see all the capital letters, waxy bronze, I forget what CL is. But these are I guess dominant, these are the recessives. You end up after this, if you were looking at phenotypes and a maze geneticist, you'd see you were just left with the recessive allele. You lost an entire arm. DS causes a breakage fusion bridge cycle. And this is what Barbara McClintock first observed, looking under the microscope at the chromosomes. She would see broken chromosomes and she'd see weird fusions, bridges. This is what's really happening. You'd have the break because DS jumped out, it's non-replicated, leaves a hole. And sometimes it'll happen on both chromatids. And non-homologous end joining, it's brawny but not brainy. Sometimes it's got 2 ends, it goes, okay, no homology, all I need is Q80 and Q70 to bind to those ends and I can put anything together. And so it'll miss repair. Instead of attaching this to that, it'll fuse it to itself. That's not good, because this right here has no centromere, and even this is not good here because you have 2 centromeres, and that means one gets pulled to one pole and one to the other, and it causes breakage. Fusion and breakage. DS is especially prone to this kind of disruptive influence on the genome. Breakage, fusion, bridge cycle, that's what it's called. Fusion bridges are formed. Dicentric chromosomes means 2 centromeres. And then you get a random breakage as you get cell division here between the 2 centromeres. This is very disruptive. Okay, so here we get finally to the story with autonomous and non-autonomous. In the case of DS, and you see DS on the right here, dissociation because it causes so much fusion breakage, but it's actually ancestrally related to activator, AC. This contains the transposase and it jumps AC. And that same transposase acts on inverted repeats that are identical on DS. So DS no longer has a transposase. And so many copies of even AC don't have functional transposases in it, but there's enough maintained to keep the family alive. But you see there are a lot of different classes of these DNA-based transposons. And you're going to need to be familiar with the concept of autonomous, which means it has the transposase, it can move itself, and any other element that contains the same inverted repeat, because that's where the transposase cuts, and non-autonomous. Genome of maize has roughly doubled in the last 6 million years due to transposase activity. Of course, man wasn't doing it. Man's only been acting on it the last 6,000 to 8,000 years probably. But in Teosinte and the ancestral species that came together to form maize, even 6 million years ago, they were increasing their transposon population. Now transposons comprise 70% of the maize genome. Have you ever eaten those transposons? Yep, you eat them every day probably. Every time you eat corn, you're eating DNA. Transposon DNA, yum. Mostly transposon, right? 70% is transposon. DS elements derived from AC, and here you see the transposase that is in these eukaryotic transposons. It looks like a eukaryotic Pol II transcribed gene. It even has introns and exons. Splice message, poly-A addition at the end, the message is capped, all of that. So here you see AC with a perfectly good transposase gene, complete with introns. But notice what the DS elements look like. They're very much shorter and really just have the inverted repeats. Shorter retain the same 11 base pair inverted repeats, non-autonomous versions of AC. A double DS element is one where DS has jumped inside of DS. It's a DS on DS crime. Here's the first one, and then there's the one that jumped inside of it. This kind of configuration is especially dangerous because it really has a high frequency of leading to breakage. DS1 elements, there's a whole family of DS, but 1 represents the extreme deleted form, contains only 300 to 500 base pairs between the inverted repeats. This generally is called a mite. So any DNA-based transposon that's just pretty much everything's been deleted in between the inverted repeats down to 300 to 500 bases is called a mite, but it's just a sort of a category. It's not a specific family. These are very common in eukaryotes, including us. Can be present in hundreds of thousands of copies. What do? They make more copies of themselves, except you make more copies of themselves. AC elements, transposition by non-replicative mechanism, cut and paste. Cut and paste, that's the cleavage and ligation, right? The second one we saw. Associated with genome replication, transposition does not occur if the DNA is fully methylated. There might be a way to control this. And there definitely is. If you can methylate the DNA that's a transposon in a eukaryote, you can inactivate it. Why is that? For a variety of reasons. One is you attract heterochromatin forming proteins, so it becomes tightly condensed and is not open to transcription. The other is you prevent the binding of transcription factors at specific sites in the promoter, a variety of reasons. But methylation is a strategy that eukaryotes employ to keep these things shut down. But they're not always successful, obviously. It's even used sometimes by bacteria. Here you see TN10, the composite transposon, which it contains tetracycline. It's also shut down by methylation. Target site is often near the donor site on the same chromosome. These things don't jump very far. If it jumps to a site that is not yet replicated, you know, you think about it, if it only makes one copy, if it doesn't make a copy of itself and just jumps, how does it ever increase its number? It doesn't, it's non-replicated. So if it jumps to a site that's about to be replicated to make two copies, then DNA replication of the host duplicates it. That's the only way. You know, on a chromosome, you've got 1,000 different origins all firing off. So it's entirely possible to have a region that's not yet duplicated jump into another region not yet duplicated, and then all of a sudden that region it just jumped into gets duplicated. Transposable are subject to epigenetic silencing. Mu. That really stands for mutator. It's a transposon in corn. The mu-DR element is often silenced when a derivative copy is inserted nearby in an inverted orientation. This kind of introduces what we hope to spend a lot more time on. This is antisense. If you have one transposase or transposon that contains a gene for transposase going from right to left or left to right if you're looking this way, and then close by you have the same transposon but it's in the opposite orientation. Because they're close by, they're going to be, the transcripts that are produced from them, the RNAs, are going to be physically very close to each other. Quite likely, they will now be able to form a duplex with each other because they're going in opposite directions. They are reading different strands of the chromosome. They're reading the same strand of the transposon, but as it goes past the transposon and gets into the other region, it'll be a different strand. In fact, when one transposase gene is transcribed by the RNA polymerase, a lot of times the RNA polymerase doesn't know where to stop. In eukaryotes, that's poorly understood how to stop the RNA polymerase. We know all about it in bacteria, but the polymerase will keep going. And so it will read the wrong strand of the adjacent identical transposon. That means you will get the complementary strand to the messenger RNA being on that long transcript. It'll start with the messenger RNA being read in the correct orientation, but the polymerase won't stop. It'll go into the next transposon copy that's facing in the opposite direction, and now as it continues on that top strand, it's now reading the wrong strand for the adjacent one, which has been flipped. So that means on this one transcript, you have an inverted repeat. That can fold on itself, and now you have duplex, double-stranded RNA. That is the substrate for lots of epigenetic silencing mechanisms. If you have duplex RNA, what we used to call antisense, we used to think that antisense shut genes down because it tied up the messenger RNA in a duplex and it couldn't be translated on ribosomes. That's why we called it antisense. It was a very obvious mechanism, didn't really need any explanation. Now we know that that's just a very, very minor part of how it works. Usually it doesn't stop translation and expression that way at all. There are whole complexes of processing, RNA duplex processing proteins that when they see double-stranded RNA, they go, aha, it must be two transposons, or it must be a virus of some sort. It's duplex RNA, it must die. It grabs it, and cuts it up into 21 base fragments. Double-stranded, 21 base fragments. Just chews it up at a very discrete length. And then it hands it off to a second protein that takes those little 21 base pairs, it separates the two strands, and it takes one of the strands and attaches it, and now uses it as a homology guide. This protein that gets this homology guide is an RNase. It chews up RNA, and now it's got an address. It's got brains. It knows what it's looking for. It's an assassin. It's looking for complementarity. It's looking for another messenger RNA that can complement that. And when it finds that complementarity, it chews and destroys the target RNA. That's epigenetic silencing, RNAi. That's used in all sorts of genetic engineering schemes these days. And it's also used every day right now in your own genome to combat viruses and transposons. So, let's see. So this double-stranded RNA that's made because you have nearby an inverted copy of the same thing. That means from that single transcript, you can form this frying pan structure, forming the duplex, which is a subject for epigenetic silencing. If they were located very far from each other, this one RNA would probably stop before it got to the other inverted copy. That's why they need to be close together. Okay, so I thought there was a whole bunch of slides there, but apparently not. So the last, I think it's the last topic here. How much time do we have? 30 minutes of glorious lecture? Okay. It's P elements. And I read the book in this section, and they have made it way more complicated than it should be. They've got figure after figure after figure on P elements. They've explained it in a way that is very confusing. So I'm going to show you their figures, and then when we get to the end of it, we're going to sit down and I'm going to explain it to you really simply. Just remember the simply story. It is controlled by two mechanisms. It's called a P element. I don't know what P stands for, but it's called a P element. It's a DNA-based transposon that wreaks havoc in the Drosophila insect genome. 30 or 40 copies of it in the genome of some of the strains that are infected with it. We're going to talk about how it's controlled, and it's controlled by two general mechanisms. One is a splicing mechanism, and the other is epigenetic silencing. So that's sort of the bottom line, and we're going to take a somewhat tortured path to get there, and at the end we'll brush off all the clutter from the table and boil it down to essential concepts. Okay, this is what it looks like. You see it has inverted repeats on each end, and it has open reading frame 1 and 2 and 3. These are really, it's the same gene. They've cut it down into open reading frames 1, 2, and 3. This makes one thing, it makes a transposase. These are the introns and these are the exons. This is all one piece of RNA. You look at that, you think there were 3 genes there. It's just a transposase gene that has 1, 2, 3 introns that need to be spliced out. When the transposase gene is transcribed into RNA, then into protein, it makes an enzyme called transposase, which goes back and binds at the inverted repeats, makes the cut so it can jump. Pretty straightforward, but already they've begun to kind of confuse us here. Length is pretty long, almost 3 kb. Terminal inverted repeats, though, these little red triangles, are 31 bases, and the actual target site that the enzyme needs is only 8 bases in length. No, I got that wrong. the direct repeats that are left when it inserts. So that's the length of the stagger when the transposase cuts the 8 bases. Got ahead of myself. There are 2 strains of Drosophila that we'll talk about. One is called the P strain, and they have what's called the P element, the P transposon. I guess the P transposon was named because they were found in the P strain. And then we have the M strain, which is naive, as they say. It's uninfected. Only about 1/3 are full length, which means the P element itself is really a mixture of transposons that are fully functional and those which now need the help of those which are still autonomous. But that's not an unusual ratio when you have a transposon. Most of them can be mutated as long as you have a healthy population there that has a functional transposase. It's only active, which means it only jumps when the male P strains are crossed with the M females. Don't even try to understand that. Here you see when a P element inactivates a white locus, it goes from red to white. So wherever it's white, that's where the P element has jumped into the I locus. So this is the early observation that led people to look at the molecular basis here. It was noticed that when doing these crosses that it had these 2 strains, P and M, and depending on whether P was male or female, you got a different outcome. If it was a P male and it crossed with a M female, you got what looked like a normal fly, but they were sterile. They'd also have other kinds of genetic disruptions. It turns out the P strain actually contains this frisky transposon that still likes to rumble, to move around. But if the male has it, it is able to jump because it has these bad phenotypes. If the female is infected and a non-infected male mates, there's no effect at all. The transposon is prevented from being active. Okay, it's not getting a little complicated, but not too bad so far. This kind of effect is called hybrid dysgenesis. Dysgenesis means it's all messed up and it only occurs in certain mating combinations. Hybrid dysgenesis. Mutations, chromosomal aberrations, one of which results in reduced fertility, often sterile. But there's no hybrid dysgenesis when the one infected or the P strain is the female. We'll need to understand that, and it'll be clear in a minute. Okay. Differential splicing of somatic and germline tissues controls the expression of the P element. P elements are only active in the germline. So there are 2 things we need to explain. What we know is that this P element doesn't jump except in cells that are making gametes, in germline cells. So normally everything's cool, right? No problem, it's not jumping. It's being repressed by whatever mechanism. But in germ cells, that's when it jumps. So how do you explain that? So it turns out that type of control is due to an error in splicing. In regular somatic tissues, which is most of the organism, there happens to be a protein, which we still don't know what it does, but it's a Drosophila protein. Drosophila has kept it all these years. It must be doing something for Drosophila. It has an RNA binding domain. Lots of proteins interact with RNA and have these domains that bind specific hairpins and loops and structures in RNA. Well, there is one that is only expressed in somatic tissues, that just by serendipity, pure accident, has affinity for intron 3 of the P element. It's just a pure accident. It sits and binds to the sequence that happens to correspond to intron 3 on the transposase. It's in every somatic cell, so that means that everywhere P element is throughout the organism, except in the germ cells, it has a pesky host protein sitting on intron 3 of the messenger, preventing it from being spliced properly. The messenger contains the intron when it should be spliced out, and within that intron, there's a stop codon. The net result is that you get a truncated copy of the protein, of the transposase. It looks like it's normally like 87 kilodaltons. This is 66 kilodaltons because it's short, because it has the intron 3 still there. Well, that transposase is not a neutral actor. It thinks it's a transposase. It knows what it's supposed to do. It knows where to bind. It goes and binds to the inverted repeat, and it chomps down, and it's like a snapping turtle it can't release. So it, in effect, acts as a repressor to the wild-type transposase. It is occupying the site, and it can't let go. That site is protected. So the transposase can never jump if one of these truncated ones binds there first. And so the book, bless its heart, calls that thing a repressor. But it doesn't tell you till like 30 pages later that it's really a truncated transposase that's acting to repress or interfere with the wild type. So forget this repressor, because they're going to call the other mechanism repressor 2 also. And they're just complicating the whole thing. So don't even use the term repressor. We'll talk about operons and bacteria and repressors, but we don't want to call that a repressor. It's just a truncated transposase that cuts but can't release because it's been cut too short. So in somatic tissue, you're going to have this intron 3 in the messenger RNA, and so wherever you have that protein, which is every cell except germline, the P element is inactive, can't jump. So that explains why in somatic tissues, no matter whether you're M strain or P strain or whatever, you're not going to see this thing active except in the germline. And why? Because the germline does not have that protein that binds to the intron. And we don't know why that is. We don't even know what that protein normally does in Drosophila. But it plays a key role here in accidentally controlling the expression of the P element. Okay, so that's half of it right there, learning the P element. Know what the term hybrid dysgenesis means? Let's look at the other situation. Can we explain this? It only works when there's a certain mating pair combination, P and M and stuff. And that's not hard either if we break it down. Activated by rapid replication and cell division. So it's activated in germline cells. Maybe they're dividing so rapidly you can't build up enough of this protein that binds to the intron. I don't know. The transposase, of course, is processed properly in the germline, so you get an active transposase and you get jumping genes. And that causes all the problems associated with hybrid dysgenesis. Okay, Now we have a series of slides that whoever did this should be doing something else. Get another line of work. Look what they've got. Repressor, and they've got arrows and dots. You know why that repressor, that's a hypothetical repressor, which turns out is not a repressor. It's simply an epigenetic silencing mechanism. Why confuse us with that? Let's go straight to the epigenetic silencing, because this to me is unnecessarily confusing and complicated. Host, repressor system, repressor, they're talking about epigenetic silencing. So I'm just gonna, I can show you all this stuff. That really means epigenetic mRNA degradation. I'm gonna show you how it works in a minute. Here you go again. It's just yada, yada, yada. Who needs it? Again, it's too complicated for me. The book thinks this is really swell. This is the lowdown. This is how it really works. Okay. In certain lines that have the P strain, for example, you have lots of copies of the P element. In some cases, 2 copies of the P element are close together. Furthermore, they are not in the same orientation. They are in opposite orientations. That means if you start making a transposase mRNA on one of them, the RNA polymerase can read into the second one, and that's going to give you an inverted repeat that this messenger RNA now can form duplex RNA, right? That's because 2 are close together and they're in inverted orientation. So on the same piece of RNA, you now have an inverted repeat that corresponds to the transposase. You have read both strands of the transposase and put them on the same piece of RNA. That means they are easily going to find each other because they are tethered to each other. They're going to immediately go together, form a duplex, and that duplex triggers this innate immunity system in all eukaryotes that makes the 21 basemer, 21 chop, and then that goes to Argonaut, and then that ends up in messenger degradation. It's easier to show with some figures here. So here you see, oh, I misspoke. I need to read my slides. It's the same principle. In certain P strains, the P element is not beside a second copy of itself. It's beside an active Drosophila gene that happens to be going in the opposite direction. So how does that form duplex? Not by the same piece of RNA forming a loop with itself, but because when it gets over here, it's the opposite strand as the transposase mRNA, and so it quickly just forms a duplex over this portion because this extends all the way over because it's in opposite orientation. So let me correct what I said before. It's when a P element lands downstream from an active host gene, but except it's in the opposite polarity as the host gene. Because RNA transcription doesn't precisely terminate in a eukaryote. It's quite common for it to read on for 1,000 bases or so. When that happens, you get a duplex potential here, and once you get the duplex, as you see there, you trigger this silencing mechanism in eukaryotes and triggers P element degradation. This is where it gets chopped by a complex called dicer, forming 21 nucleotide fragments. They start out as double-strand fragments, but they are quickly separated by the next enzyme that it's handed off to, which is called argonaute. Argonaute is the RNase that cuts, but it's guided by this guide RNA produced from the duplex area, which was formed from transposase mRNA. So this RNase called argonaute is looking for transposase messenger RNA to complement, recognize, and now cut and degrade. This is a basic mechanism of RNA silencing in eukaryotes. You get a duplex, which is degraded by dicer complex into 21-mers, and the 21-mers are split into their separate strands, and each strand will bind to an argonaute protein, which now is an RNase with a little information module with brains. It's on a mission to only cut RNA that is complementary to its little guide RNA here. That's in we have that happening right now. That is protecting us, keeping our transposons asleep, and protecting us from RNA viruses. It's called epigenetic silencing. Transposase RNA, messenger RNA, of course, is the target because that's what it was originally made from. And that, of course, is degraded, no gene expression. That is the end of today's lecture. Yeah. Who said I can't talk fast? Are there any questions? When is the homework? When's the bioinformatics due? How's it going to get graded? If I make a mistake, can I get it corrected? Do I submit it through the regular assignment site, or can I just email you? You can take a copy of it ahead of time. Take a look at it. Okay, I'm glad someone asked that question. The bioinformatics project is active now. The first part's in 3 parts. You get a chance to submit it more than one time. I have a team of very generous TAs. They want to allow you and make it possible for you all to get A's. You can submit this multiple times, of course, until after the due date. If you submit it early, it will get graded, and if you don't have it perfect, there'll be comments, and you can correct it and resubmit it, and you can get the full points on it. The Bioinformatics Part 1. There are a series, you know how to get to it? If you go to the website, there are a series of little circles on the right. One of them has a flask. The course designer did it. I had a great big picture of a piece of DNA and stuff, and she reduced it to a little icon. It's the last one, and you hit it, and a little thing will pop out, and it'll say Bioinformatics Project. Click on that, it takes you to a whole page that has instructional videos and everything. If you have a question, go use the course. Don't email the TAs directly, but do it all through the course, through the inbox down there. They are sitting at home right now ready to receive those emails. They love to give helpful suggestions. So that's how that's going to work. And then we'll do the second part the same way, and then the third part. It'll start you off very simple. This is all simple bioinformatics. You'll start by looking at FASTA files, and it'll eventually get you into the databases where you can go into the human genome and look at any piece of DNA and do whatever you want to in your garage on Saturday with it. That's the point. Not only GMO orange juice, but GMO. So, okay, any questions? Can we take exam 2 by live proctor? Another excellent question. I'm in the process, hopefully I'll do it when I get back. I'm going to put up a sign-up group like before. There's not enough time and space for everybody to have that option, but I'll try to cycle through 60 to 80 people. That's about all. And Monday night, of course, the last night, that's when everybody takes it. So it'll be Monday night when the exam opens, the last Monday, it opens on, I think, is it Friday or Thursday or Saturday? It runs for about 3 days. Closes Monday night, though, always. External camera, do you need an external camera? Yes, unless you cheat the system by coming over there and have me look at you like this. So what kind do you get? Well, some people had trouble with external disc. If you have an external disc, they're usually a little mini disc. And you got to get an adapter before you can put that in there. The camera that I recommended, and I just stumbled across it, I don't know ultimately what the best camera will be, but it's a 1080p. It has a manual focus. So you do it yourself until the image is not blurred. So that means you don't have to have drivers and stuff to be able to control that, so it's real simple. And the one I have on my Mac, it didn't take any driver or anything. I just plugged it in. You can't see a thing unless you activate the camera through an existing app you have, like Skype. That's how I got to see whether my camera was working. I went to Skype and chose that external USB camera to look through, and sure enough, it works fine. ProctorU has their own app. They don't need you to have any apps at all. They will know how to access it. Where should you position it? Well, the diagram has it on one of the distal edges of the desk at a 45-degree angle. That's what the ProctorU representative for UF told me. But playing around with my little camera, that seems silly because my computer's in the way. I found that if I position it beside me, so I can see the screen and see my hands, then you can tell where you're looking. I don't have to see where your eyes are because it's very obvious where your head is. And I can see, in fact, I can see what you're looking at. So beside you is best from my point of view when I go look at these videos. One way to do it is take a chair, turn it like this, and clamp it onto the back of the chair and have it look at you from the side. Another way is to put it on a stack of books, but ProctorU has this silly rule where you can't have books, even if they're closed, and even if the only way you could open them was to take the camera up like this and open the book. They still don't like a book. I think that's crazy. I would say a stack of books is fine as long as they're not open books. But so that's why maybe a chair sitting beside you or something as something to clamp on might work well. If you have it elevated from the desk surface, it gives a better view of the keyboard and everything. Because now when you look, the hand looks like a big mountain. And you can barely see over it. But if you're up a little bit, you can see down into the valley, and the keyboard and everything looks cool. So some of these things you can get little tripods, but that's why I say you can just put it on, a milk carton cake or a cardboard box, something you can clamp it on to. And look, you can be inventive. What you're trying to do is to make an excellent case that everything's on the up and up. You're trying to convince me, nothing to see here, move along. And the best way is just give me a clear view. I'm going through these videos and I don't look at every frame, but I've seen enough where I can sort of tell whether the person is nervous and looks like they're in trouble, and then I kind of slow down and watch them answer a typical question they don't know the answer of. And they don't look right at the beginning because the proctor's still awake. I'll go like 3/4 quarters through and you're getting comfortable with there's nobody watching. Oh yeah, kitty, kitty, kitty. And the cat, of course, has the answers on his back. And the little cat. There's a guy in the next room with the, okay, so there are all sorts of schemes. And what I can say is from looking at this class that my suspicion level is near rock bottom. You guys apparently are just above board, outstanding citizens, every one of you. But I will continue to monitor, but I would prefer that we stop all the funny business just by deterring it. That's the best way. And I'm relieved to know that most of you, in fact, I'm having a hard time finding any rascal among you, and you've got to be out there. Maybe I'm losing my touch. But I'm impressed that very low incident rate this semester. It looks like things have really turned around.
Will you still offer the service where we can go in and take it in the microbiology building?
Absolutely, with great reluctance. I can only take a certain amount because just the time and space and all that stuff can still operate in that universe. But I put up a sign-up group. Hopefully I'll remember when I go back in a few minutes that you can sign up, first come, first serve. If there are people that have emergencies of one sort or another, just show up and I will make accommodation for you. You can get work done.
You still have the cameras that we can check out?
Yes, I do. I sent out 10, I got 9 back. That's a lot better than I figured. So I do have cameras for checking out, and I'm happy to check those out. They seem to work fine.
I condensed all your slides and took the blue outs. Do you want me to send those to you?
Oh, yes. And how do you spell your name? Yeah, if you could send me that, that'd be swell. And if any of you have used a camera, because it takes so long to go through these, I'm making my way, and I find only 1 in 4 of you actually used a camera. I'm having trouble getting a big collection of configurations that I can go to ProctorU and give them advice as to what they should tell, and what advice I can give to other people as to the kind of camera you should use. If you could send me through the inbox, just put the message, say camera, tell me what camera, your brand, what you got. Is it a 1080p or is it 720p? That's pretty much all I need to know. If you got something about how you configured it, you said you set it on the back of a chair or you set it behind, or if you got any comment about the Proctor was horrible, you can tell me that too. I certainly believe all that. I'm going to provide you an opportunity for feedback so I can learn how ProctorU is going and how the camera thing is going. I think they're kind of 2 issues, and I've seen enough of these videos where I know ProctorU is sometimes just takes forever, and then they come on like robots and just can't be reasoned with at all. And it's just, and some people hold up their ID, but they hold it too close to the camera, especially the external USBs. And the Proctor never says, if you just back up, sir, like about a foot, it'd be in perfect focus. No, the Proctor never says that. The guy keeps getting it closer and closer and closer and closer. Pretty soon you can see right through it. And I'm going, move it back, move it back, move it back. People start looking in that little window of my office. He's looking, he's yelling at the computer again. So, everybody happy, right? And that's all I have today.